All right, everyone, thanks for joining me. This lecture will cover the diversity of prokaryotes and eukaryotes that are found in the microbial world. First up, we have Wolbachia pipientis, which is a member of the bacteria. Wolbachia pipientis is a gram-negative pleomorphic bacterium which is found as an intracellular symbiont in both arthropod and filarial nematode worms. In the nematodes, it seems that it's primarily a mutualist. It benefits from the relationship, as does the filarial worm. However, in most arthropods, the Wolbachia bacteria are considered parasitic. So you can see here, there is a picture of a wasp egg, and all of those little glowing circles are Wolbachia bacteria that have found their way into that wasp egg. The really interesting thing about Wolbachia when you're talking about it in relationship with filarial diseases such as elephantiasis and African river blindness is that if you kill the Wolbachia using antibiotics, you kill the worm and you help treat the filarial worm infection. Now, in elephantiasis, that's a successful treatment. However, in African river blindness, the treatment, which kills Wolbachia and then kills the worm, actually is what causes the blindness associated with this infection. So that's not therapeutically useful. What we're going to focus more on is the use of Wolbachia to stop mosquito-borne illnesses such as malaria and dengue fever. Before we move on to talking about how Wolbachia can be used to treat mosquito-borne illness, just a note about these filarial diseases of elephantiasis and African river blindness. You can see at the top of your screen there a person whose right leg is really swollen and distended, and that's actually the result of this filarial disease. The worms migrate to lymph nodes, in the case of this person here, most likely the groin, and they start to reproduce in the lymph nodes, and they get so compacted in the lymph node that they block all of the lymph fluid from the lower extremity, and it accumulates in the tissue, causing this edema that you see here in elephantiasis. On the other hand, African river blindness, as you can see here, though the worm is inhabiting the eye and there's redness and irritation, it doesn't cause issues until the worm dies, at which point it damages the eye and causes blindness. All right, having said that, let's move on to treatment with Wolbachia of vector-borne illness. So how is Wolbachia used in order to treat these mosquito-borne illnesses? And the answer lies in the fact that mosquitoes, for the most part, cannot be co-infected with a virus when they have already been infected with Wolbachia, and also due to the fact that infection with Wolbachia affects the fertility of mosquitoes. If you look at the left-hand side of this diagram, you'll see that that's a mating between an infected female and an infected male, and infected there means infected with Wolbachia. When that mating occurs, the eggs will hatch and they will produce infected mosquitoes, meaning mosquitoes infected with Wolbachia, which of course will not be able to be infected with either malaria or dengue fever. If you have an infected female and an uninfected male, the eggs again hatch and you have infected mosquitoes. So what ends up happening in both of those situations is that you end up with a population of mosquitoes which, because they are infected by Wolbachia, will not pick up malaria or dengue fever. The problem with this scenario is that you still have mosquito biting going on. So you still have the pest aspect of the mosquitoes, you just don't have the disease aspect of the mosquitoes. Now, on the third line here, we see an uninfected female mating with an infected male, and that actually leads to eggs that will not hatch. So this scenario both takes care of the number of mosquitoes and the ability to transmit disease. So fewer biting incidence and also lower incidence of disease. So they have used both the release of Wolbachia infected females and Wolbachia infected males as a treatment for disease. And it really just depends on how common the diseases are in these areas as to what particular strategy is going to be used by epidemiologists trying to control the spread of the disease. So just to make that point again, the uninfected female who has no Wolbachia will bite a human who is infected with dengue virus. That dengue virus will be taken up into the mosquito's body. It will replicate within the mosquito's body, and then when the mosquito takes her next blood meal, she will inject those dengue fever viruses into the second host, thereby transmitting dengue fever. Likewise, she would also transmit malaria. Case scenario number 2, a female that has been infected with Wolbachia will bite a human. She will take her blood meal. She will pick up dengue virus from that person if they are infected, but because the virus cannot replicate alongside Wolbachia, the dengue virus does not spread when the mosquito takes her next blood meal. So this becomes a really interesting way of treating these mosquito-borne illnesses. All right, next up we have another bacterium, Rhodococcus ruber. Rhodococcus ruber is very important because of its ability to bioremediate several different kinds of environmental pollutants. And you'll remember from discussions of earlier chapters that bioremediation means the cleaning of toxic products in water, in soil, in the atmosphere, wherever it might be, by the use of microorganisms. So bio, because this is done by a living organism, remediate to clean up. And the reason these beautiful reddish orange microbes are so good at this bioremediation is because they are very fast growing. They grow at a neutral pH and they grow at a relatively easy temperature to maintain. So they are neutrophiles, they are mesophiles, and they are fast growing. They also can fix nitrogen, which is kind of fun. They are involved in the desulfurizing of fossil fuels. They are capable of metabolizing hydrocarbons like crude oil and other fossil fuels and using those as an energy source. So they're good for cleanups of crude oil spills. They're able to accumulate heavy metals out of the environment so they can detoxify soils. They also bioremediate a number of environmental pollutants, and they're able to degrade organic pollutants and pesticides. And one of their, I think, coolest attributes is that they're actually able to eat styrofoam and decompose it completely. So you eat a cheeseburger, they eat styrofoam, and they can also do that with phthalates. Both of those things are a big problem because they're very difficult to work with and get out of the environment. So our ruber is very important for its bioremediation characteristics. Next up, we switch gears and move over to the other group of prokaryotes, the archaeans. First up, we have a member of the Eukaryotic, Halopharax volcanii. And as you can see from the picture up there, we have a bacillus, which, because it's an archaean, is always going to be gram-negative. Remember, archaeans, though they are prokaryotes, lack peptidoglycan in their cell wall, and so will not ever retain that primary stain. This guy is an aquatic, halophilic mesophile, which is easily cultured, and because he's easily cultured, he's become the model for understanding archaeans since he was isolated from the Dead Sea in 1975. And as they've studied him, they found that the same horizontal gene transfer systems that are present in bacteria are also found in this archaean cell here. They see evidence of a natural mating system between members of H. volcanii, which is cool. Next up, We're keeping with the archaeans, but switching gears and moving on to Crenarchaeota with Sulfolobus sulfatericus, our next archaean example. All right, S. sulfatericus is an aerobic, thermophilic, acidophilic, terrestrial, volcanic hot springs inhabitant, which has the really cool ability to either take sulfur out of the hot spring and grow chemoautotrophically with it, or to grow chemoheterotrophically using any number of organic molecules that it finds in its environment. This guy's been found all over the place in Yellowstone, in Mount St. Helens, in Iceland, in Italy, and in Russia. And very interestingly, rather than using NADH as an electron carrier, which is moving electrons to the electron transport chain, S. sulfatericus uses ferredoxin instead. Next up, we move on to the eukaryotes, beginning first with our fungal organisms, the first of which is a basidiomycota called Cryptococcus neoformans. Now, just because we've moved on to the eukaryotes doesn't mean we've moved fully over to this idea of multicellular organisms. In fact, as you can see from the light micrograph at the top of your screen, this is a spherical yeast. This is a member of the fungal kingdom, which is unicellular. It is relatively large, and it does have a capsule that protects it in the environment. As a member of the basidiomycota, it forms club-shaped basidiospores, which are shown in the transmission electron micrograph at the bottom of your screen, releasing spores. It was discovered growing on peaches, but it turns out it has a worldwide distribution all over the place in soil, decaying wood, tree hollows. It also is found in bird droppings. The interesting aspect of Cryptococcus neoformans growth, though it is found worldwide, it only grows between 37 and 39 degrees Celsius. Now, the reason we're going to talk about Cryptococcus neoformans here is because it can be an opportunistic fungal pathogen. So, opportunistic meaning a person typically has to have a reduced immune function in order for this to cause a problem, because it turns out most people are exposed to Cryptococcus neoformans during childhood and are asymptomatic. What might happen is that they might have a reactivation of that cryptococcal infection that is symptomatic if they become immunocompromised, or if they are exposed to Cryptococcus neoformans as an immunocompromised individual. What happens is the Cryptococcus neoformans spores, the spores are inhaled and they can cause pneumonia. In an immunocompromised individual, that pneumonia can very commonly and very quickly move to the meninges and cause cryptococcal meningitis. So this is an infectious disease that is non-communicable, meaning that a person with cryptococcal pneumonia will not transmit that pneumonia to people around them. If you contract cryptococcal pneumonia, your primary symptoms and signs are going to be cough, shortness of breath, chest pain, and fever. If that infection moves to the central nervous system, the symptoms will include headache, neck pain, light sensitivity, confusion, behavior changes, again, fever, nausea, and vomiting. So severity of signs and symptoms increasing, obviously, when you end up with a central nervous system infection. This cryptococcal disease is diagnosed via a latex agglutination test, which is something that we'll be talking about in Chapter 18, where antibodies for Cryptococcus neoformans are looked for in the body. It can also be cultured from sputum samples, from tissue samples, or from a bronchoalveolar lavage from a patient. These obviously can be very invasive, and so a lot of times the latex agglutination test, which we're going to learn in Chapter 18 is an indirect way of looking just from a blood test, is a bit easier diagnosis. If you have a confirmed diagnosis, you will be started on fluconazole, amphotericin B, and flucytosine, because the central nervous infection has a 44% mortality rate even with treatment. All right, moving out of the fungi and into the algae, we have our first algal species, Chondrus crispus, which is a member of the red algae, the Rhodophyta. And finally, we get to the macroscopic organisms. This guy happens to be found along the rocky Atlantic coast of Europe and North America, and its common name is Irish moss. As you can see from the picture there, it is macroscopic. It grows to about 20 centimeters in length and grows from a holdfast and grows into these pretty little fan-like projections that you see pictured here. Like many algal species, C. crispus exhibits what's called an alternation of generation, where there's both a haploid and diploid form of the organism. The haploid form is shown up at the top left-hand side of your slide, and the diploid form is shown at the bottom right-hand side of your slide. Fun fact about C. crispus, when it's dried, the powder that results can be used to produce thickening agents such as the agar that we use in order to make solid media in the lab, and as a food additive called carrageenan. So, in the lab and in your grocery stores, C. crispus is used as a thickening agent. Next up, Thalassiosira pseudonana, a member of the Prokaryotic, which, as you can see from these pictures, are very pretty. Prokaryotic are diatoms, and they are non-motile unicellular algae which come in all kinds of shapes and sizes, and all of them have in common that they are surrounded by a cell wall called a frustule, which is made of pectin and silica, so it's very glass-like, and that's what you're seeing in that top picture there. As I mentioned, they're morphologically diverse. You can see that from the pictures here. They come in long pin-like forms called pinnate diatoms. They come in round forms called centric, and they also form filamentous chains. When conditions are right, they form blooms, and the fossilized remains of diatoms are used to make diatomaceous earth. So this guy right here, Thalassiosira pseudonana, was chosen as the first eukaryotic marine phytoplankton to have its entire genome sequenced. So you're looking right here at a beautiful centric phytoplankton celebrity. Now, as a diatom, this organism has a really interesting problem. Because it has that rigid cell wall, it actually unlocks the two halves of the cell wall as it undergoes asexual reproduction. And as it does that, one member of the pair will end up smaller than the original diatom. And as you can see, as you move from left to right across this figure, they get smaller and smaller and smaller, and they get to this point of no return where the cell decides, no, we've gotten too small, and then it will switch from asexual reproduction to sexual reproduction, forming gametes that fuse and then go back to the original size of the organism. All right, on to another of our algal species. This one, though, a member of the Dinoflagellata Noctiluca scintillans, which as a member of the dinoflagellates is one of over 2,000 described species, which are incredibly diverse ecologically. Some of them are mixotrophs, able to move between autotrophic growth and heterotrophic growth. Some of them are toxic, so they're pathogenic in the aquatic environments where they live. Some of them are involved in cloud formation because they produce DMSP, which is converted to DMS and is involved in cloud formation. And they're also really important for coral reefs. They actually live inside of coral reefs and provide a very important symbiotic relationship there. In addition to the coral reefs, there are also symbiotic dinoflagellates in different species of jellyfish. So all over the map in terms of where they are and what they do. The species we're talking about here today, N. scintillans, is the sea sparkle, which tends to bloom after the phytoplankton because it eats them. So that diatom that we just mentioned, if it blooms, this guy will come along afterward and eat that diatom. And the really cool thing about this sea sparkle organism is that when it is agitated, as it might be when waves occur or when water moves or when you jump in the water, it bioluminesces, it excretes a chemical that glows in the dark. Now, this particular organism is non-toxic, though there are some species of dinoflagellate that do produce neurotoxins. However, it's not completely a non-issue because it does produce high levels of ammonia as it feeds, and it also may contribute to the neurotoxicity of other algal blooms. All right, on to the Ciliophora. Stentor roselii is a complex protozoan which is large, ciliated, and contains multiple nuclei. It feeds using cilia and is primarily found in fresh water where constant osmotic pressure is avoided by the use of a specialized organelle called a contractile vacuole. S. roselii have a really interesting morphology. They have given up being motile and have become sessile, attached to the environment, and instead have a ring of cilia that beat and create a current in the water that draws food toward the organism. So they actually use their cilia to feed rather than to swim. And they're fairly large. They're about 1 millimeter in length, so they would be visible to the naked eye, though obviously not with this level of magnification. One thing about ciliates that has been discovered and is kind of interesting is that they undergo a process of conjugation. Unlike bacteria, you'll note as you watch the micronuclei come together from two different ciliates through this process of syngamy, both partners receive genetic information from one another. So rather than there being a donor and recipient cell, both cells act as donors and recipients. So at the end of the process, there in 7, 8, and 9, you have 2 recombinant cells as a result of this conjugation rather than just one. All right, next up we have Giardia lamblia, which is a member of the Archaeozoans. You might remember from lecture material that the Archaeozoans are flagellated eukaryotes, but they contain no mitochondria. And this Giardia lamblia is the causative agent of what's commonly called hiker's diarrhea. And you can see its 2 forms on the right-hand side of your screen, the trophozoite form, which is motile and active at the top, and then the cyst form, which is what is transmitted during infection because as a cyst, it is highly resistant and remains in the environment for long periods of time. And Giardia was actually discovered by Antony van Leeuwenhoek. It was one of his animalcules that he saw under his microscope. So when you ingest a cyst, your digestive tract actually digests away the cyst coating and frees the trophozoite inside. And once they're inside the gastrointestinal tract, the Giardia will adhere to the intestinal wall using an adherent disc. And once there, you may have a completely asymptomatic infection, in which case you would be transmitting disease to other people without having any idea that you yourself were sick. If you are symptomatic, which is also common, symptoms would include stomach pain, gas, bloating, and diarrhea. And if you are diagnosed with Giardia lamblia, the good news is it can be very easily treated with flagyl, though it is resistant to conventional water treatment. So it's one of those indicator organisms for successful water treatment, and it is very common in lakes and streams in the United States. So this is one that you need to watch out for because its host is not only human beings, it's found in beavers and dogs and wolves and coyotes and all sorts of other organisms, which may also be depositing cysts in their fecal material into the environment, which finds its way to water sources. So clean water, always important when you are out hiking or you might contract hiker's diarrhea. So how do you get this? Again, you ingest a cyst either from contaminated water, from your hands, from food, from a fomite. That cyst is digested in your stomach. The trophozoite moves to the intestinal wall where it adheres, and they mature and multiply via sexual reproduction or asexual reproduction. Some of them then form into cysts, and both the trophozoites and the cysts are passed in the stool, but it's the cysts that persist in the environment and are capable of causing infection in another host. If another host were to take up a trophozoite, it would be digested in the stomach and you wouldn't get infected. All right, Plasmodium falciparum, the apicomplexan. Plasmodium falciparum causes the mosquito-borne illness malaria. It itself is a unicellular protozoan parasite which is non-motile, and P. falciparum is the deadliest species that is able to cause malaria. There were about 219 million malaria cases in 2017, many of them in children under 5 years old. This accounts for about 61% of the deaths, and every day 2.4 billion people are at risk of malaria, which, as I mentioned, is transmitted through mosquito bite. You can see here the multiple stages of P. falciparum inside of the red blood cell where it lives as an obligate intracellular parasite. And the symptoms of malaria are actually caused because as this P. falciparum grows, it ultimately bursts the red blood cells, and that's what causes the symptoms. So you'll get these cyclic fever and chill episodes anywhere from every 6 hours to every 24 hours, depending on the species of Plasmodium you've been infected with and how well your immune system is combating it. But every 6 to 24 hours, you'll cycle between fever and chills. You'll also have headache, dizziness, nausea, vomiting, diarrhea, increased heart rate, jaundice, low blood pressure, and enlarged liver and spleen. You may also suffer the complications due to the red blood cells getting really sticky, because when they get sticky, they obstruct microcirculation and they can cause organ damage and cerebral malaria. So malaria is transmitted through the bite of a mosquito who injects sporozoites from its saliva into the body, which migrate to the liver, where they undergo part of their life cycle. They become hypnozoites in infected hepatocytes, and then those hepatocytes rupture, releasing merozoites into the bloodstream, which attach to red blood cells and enter them and go through these trophozoite stages and then ultimately become gametocytes. The gametocytes are taken up by a second uninfected mosquito in whom the life cycle continues until the sporozoites are formed, which then burst through and get into the salivary glands of that second mosquito and are transmitted to a person during a blood meal. The treatments for malaria, even though people didn't know what caused it, have been around for a long time. Initially, there was something that was found to be useful, and that was a tincture made of the bark of the cinchona tree, and then quinine, and then artemisinin from Project 523, which was a result of Viet Cong troops were dying at actually a greater rate of malaria than they were dying of battlefield wounds. There is currently no vaccine for malaria, and it is a big problem in places where these mosquitoes are prevalent, and these mosquitoes are becoming more prevalent all over the world. Next up, we move on finally to Kingdom Animalia with some fun worms, including your friend and mine, Dicrocilium dendriticum, which is a liver fluke. As a member of the Trematoda, this guy has a very complex life cycle. It is commonly known as a fluke, which is a flatworm. It has two suckers. It is a hermaphrodite, and it is protected in the environment by a hefty skin called a tegument. And just to drive home the point of that complex life cycle, if we start at number 1, we have our adult fluke. Move to number 2, we have an egg, a myricydium, a sporocyst, cercarii, slime ball, and then finally a transformation to a metacercarii. So we said in lecture, what's the kicker about these worms? They are characterized by their complex life cycles, which in Dicrocilium dendriticum, we can certainly see that happening. In addition to a complex life cycle, Dicrocilium dendriticum also has a complex life. It requires 3 different hosts in order to complete its life cycle, and they include cows, ants, and snails. The cow ingests the metacercaria, which has been inside of an ant, which we'll talk about in a minute. So cow eats grass. In that grass, there is this metacercaria, which moves into the cow's digestive tract, into the liver, and then it undergoes these transformations, sexually reproduces within the cow, and then releases eggs into the bile duct, which move into the gastrointestinal tract and are ultimately passed in the stool, which is consumed by the snail. And inside of the snail, a myricydium hatches from the egg. It becomes a mother sporocyst, which becomes a cercaria, and that cercaria becomes flagellated and actually moves through the snail and is secreted along with the slime trail that the snail leaves, which is eaten by an ant. And here, if it wasn't weird enough already, is where things really get weird. So this cercaria encysts and becomes this metacercaria, and the metacercaria move into the ant brain, which granted is not much, moves into the ant brain and sort of hijacks the ant and has the ant stop everything it's doing, move to the tip of a blade of grass and sit there in the cool morning. And if it's not eaten, it moves down and hides during the heat of the day, and in the cool of the evening, the ant climbs back up, waits again for a cow to come and eat it, and the ant will repeat that cycle over and over and over again until it dies or the cow consumes it. So once that metacercaria gets inside of the cow, it hatches, it grows into an adult, which undergoes sexual reproduction and produces those eggs in the cow. So for those of you who were curious, the cow is the definitive host of Dicrocilium dendriticum because the cow is where sexual reproduction occurs. That's a normal fluke. Now we get to look at a blood fluke, and in this case we're talking about Schistosoma mannsonii, which is one of several species of schistosoma, which are dioecious, meaning they are male and female. They are parasitic flatworms, and as you can see from the picture at the top of your screen, they are sexually dimorphic. The male is much larger. He has a groove along his ventral surface in which the much smaller, skinnier female will reside. They have digestive and excretory systems, and it is estimated that there are about 210 million people infected with schistosoma today. Though it is treatable with praziquantel, it is really isolated primarily to the African continent where these worms are common, and without treatment it can lead to malnourishment and death. It has 2 hosts, not 3 like Dicrocilium dendriticum. This one infects snails and humans, and it is released in the fecal material of human beings and then infects intact skin in its next host. It causes rash, fever, muscle aches, and it can also lead to bladder cancer, liver fibrosis, damaged organs, and developmental issues in children. Schistosoma lives part of its life cycle out in human beings and in snails. In the human being, the adult schistosoma blood flukes undergo sexual reproduction, so human beings are considered the definitive host, while the snails are considered an intermediate host. However, a lot of eradication efforts have been made toward trying to get rid of the snails so that the life cycle of schistosoma is interrupted. So the adult worms live in the hepatic circulation, and they get there within about 15 days of infecting a person, and if they find a mate, they will sexually mature and migrate as a pair to the mesenteric veins where they will produce eggs, and those eggs will be released in the gastrointestinal tract and the urinary bladder. The eggs hatch in the water. They form myricidia, which are capable of infecting snails, and the myricidia inside of the snails undergo this transformation to cercaria, which are able to attach to and penetrate intact skin in the next host. And these cercaria are able to live for about 12 hours outside of the snail, and they wait until light, and then they swim toward people, and some of them, it's thought, are able to pick up on chemical signals in the water and follow those chemical trails toward people, at which point they will burrow through the skin and make their way through the skin to the lungs, to the heart, and ultimately to the hepatoportal circulation. When a person is infected with schistosoma, because it's able to burrow through intact skin, you'll typically get a painful blister at the site of infection, which a lot of times is in the lower extremities, as you might imagine if you're walking around in the water. Schistosoma recently has been implicated as a comorbid fact with HIV infection because these ulcers can also occur in the female genital tract, and when they do, that broken epithelial layer in the vagina makes a much easier transit for the HIV from the semen of an infected male. So there's a lot of research being done in order to see how we can reduce the level of HIV transmission by also reducing the incidence of schistosoma. Now, because these blood flukes drink blood, they significantly affect the health of their host. People have anemia, and people can become so anemic that they are malnourished because it's not just one pair of these worms that's living inside of a person's body. As you can see from distended abdomen of the boy in the front of this picture, the abdomen is very distended because of the large number of worms, and his limbs and face are very gaunt because he is not getting enough nutrition from his food. It's all being stolen by these worms. If you were looking for a little good news at the end of that organism, I hate to break it to you, but you will not be finding that yet. Now we move on to the tapeworm, Tinea solium. Tinea solium is commonly known as the pork tapeworm, and it is a hermaphroditic parasitic helminth which infects, as its name suggests, pigs but also humans. It can live for up to 25 years in its host and grow up to 10 meters in length. The ingestion of larvae from infected meat causes gastrointestinal disease. Those larvae are shown with the yellow circles. And ingestion of eggs causes cysticercosis and neurocysticercosis. We will start with the pig. So the pig eats the eggs or gravid or lost proglottids, pieces of the tapeworm, typically in fecal material, right? Pigs are not super picky about what they eat, so they will eat fecal material. And once they do that, those eggs will move into their intestinal tract, they will hatch, and they will move into the musculature of the pig by actually penetrating the intestinal wall. Once they do that, they will form larvae in the muscle of the pig, and then if the pig muscle is consumed and has not been cooked properly, a person will contract the larva and it will go into their intestinal tract where it will mature into this adult worm, which attaches itself to the intestinal tract. That's where it can live for 25 years and grow to 10 meters in length. And as it does that, it's constantly stealing nutrients from the gastrointestinal tract, causing weight loss, also because of its size, causing abdominal pain and other gastrointestinal issues. So this is just intestinal infection with a tapeworm. Now, what can happen in human beings is something called autoinfection. And during autoinfection, a person will actually autoinfect or infect themselves through oral fecal transmission of the eggs or the proglottids from their own fecal material because they're not washing their hands well, or because they're a small child who happens to touch those parts of their bodies without washing their hands. And when you do that and you deposit those eggs into your own mouth, they will do just what they did in the pig, and those eggs or proglottids that you've ingested will hatch, they will penetrate the intestinal wall, and they will circulate into your musculature, and worst case scenario will migrate into the brain. So if they migrate into your musculature, that's cysticercosis. If they migrate into your brain, that is neurocysticercosis. And it's typically not a one or the other. It's typically cysticercosis and then also neurocysticercosis. Case in point, we have a 21-year-old male who presented to the emergency room with severe headaches and seizures. He had multiple small subcutaneous nodules all over his body. They were palpated in several areas, including his chest and legs. They did a head CT and found dark areas that were consistent with cysticercosis. And when they did X-ray films, those X-rays identified many calcified lesions in the muscles of his legs, which are shown in this X-ray here as these almost looks like little white lines all over his legs. They were also found in his chest and neck. They then ordered an echocardiogram, which showed calcified lesions on his heart. He spent about a month in the hospital on antiparasitics to remove these worms from his body, and he was discharged doing well with anti-seizure meds. He also, incidentally, was not found to have intestinal infection with tinea. He must have gotten the eggs from somewhere else and ended up with cysticercosis and neurocysticercosis. This is, I think, the grossest of the parasites. And I know you're thinking, how can it get worse? Just wait one moment and you will see. Here we are going to talk about the threadworm, Strongyloides stercoralis. He is a nasty, nasty worm, which is transmitted in the soil as either male or female, and he is a parasitic nematode roundworm who is about 1 to 2.5 millimeters in length, so he's visible with the naked eye. He is capable of inhabiting the small intestine of human beings, primates, cats, dogs, all over the place, found in tropical and subtropical climates. His modus operandi is to penetrate intact skin and make his way to the small intestine, but sometimes he gets lost and you end up with lost little worms all over the body underneath the surface of the skin, which are shown in this picture at the bottom of your screen as these red wiggly lines. Those are actually worms sitting underneath the surface of the skin, wondering where the gastrointestinal tract is. Now, if they make their way to the gastrointestinal tract, they may be asymptomatic, but you can also have gastrointestinal and respiratory issues, which may seem strange, but just hold on to your hat and we'll explain why you can have both respiratory and gastrointestinal tract issues. They are diagnosed from a stool sample or an antibody test from the blood, and when you are diagnosed with strongyloides, they are treated with antiparasitics. So here is what strongyloides does. So let's start at the bottom, number 1. So the larvae in the intestine are secreted in the stool. They develop into free-living worms. They cruise around, they find a mate, they make some babies. The babies hatch into larvae, and they become these infective filarial worms. And they penetrate intact skin, initiating the infection. So these are common in the soil, so walking around with no shoes on allows them to burrow into your skin. And there, from your foot, this worm has the big job of trying to get into your circulatory system. So it burrows, gets into a capillary, moves into the circulatory system, moves through the circulatory system into the lungs, where it punches into the alveolar space and moves through the airway, through the bronchioles, into the bronchi, into the trachea, into the pharynx, where, and this is where I think it's really disgusting, he jumps out of the pharynx and causes a little tickle in the throat, which causes you to clear your throat and then swallow, at which point he finds his way into the gastrointestinal tract, which was his goal all along. He then moves to the small intestine where he becomes an adult. Adult female worms in the intestine release eggs. Eggs are hatched, migrate to the lumen of the intestine, and then are excreted from the intestine into the environment. So here's the double bummer. You can get an autoinfection with these strongyloides as well, right? So they become filarial in the large intestine, and they can penetrate the intestinal mucosa and follow the normal infective cycle within the body, right? They can also do that outside of the body. So you end up with these terrible multiple infections of this organism. And if it's not clear already, the reason why it can cause both gastrointestinal and respiratory tract issues is because it can cause damage to the lungs. You can have a buildup of these strongyloides in your lungs, and that can cause difficulty breathing, causes coughing. There have actually been cases diagnosed because a person has a persistent cough, and upon examination, their lungs have lots of strongyloides inside them. So that causes the respiratory issues. If it's just doing its normal thing in the gastrointestinal tract, it can cause issues for you there as the body tries to deal with strongyloides. Okay, now this is going to be gross, but I'm just going to tell you we're going to end on a pleasant note talking about Dracunculus medianisis, which is commonly known as the guinea worm. The guinea worm is a very large sexed worm, so there are female worms, there are male worms. It's rarely fatal, but it has a huge economic impact because it makes it difficult for people to get around and do their jobs. It's a very fertile organism. Females can carry about 3 million embryos, meaning that a single infected person can release 3 million infectious agents into the water. It has both human beings and a microscopic arthropod called a copepod, which is kind of like a microscopic crab, as hosts. It has about a year-long disease cycle from the time that you are infected until the female is capable of releasing those eggs. And she does that by releasing them through a blister that forms on the skin. As I mentioned, it is non-fatal, but the rupture of the worm can cause a severe allergic reaction, which means that you can't just yank this thing out of yourself. You actually have to remove it very slowly over a period of a few weeks by grabbing the worm as she tries to release her embryos, tying the end of the worm to a small stick like a matchstick and slowly twisting out a couple of millimeters of this worm every day until she's gone. So while you do that, you can take anti-inflammatories, you can take antibiotics to keep that blister from getting infected, but if you try to take an antiparasitic to kill the worm, you can cause severe allergic reactions because the worm dies and bursts in the body. So here's the good news. Access to clean water, which is super easy because all you have to do is drink through a straw, is moving this infection toward eradication. The guinea worm is a great candidate and will be eradicated sometime soon throughout the entire world. And that is primarily due, as I mentioned, to the WHO, the World Health Organization, and other organizations passing out these straws, which these two little boys are shown holding on to, which they drink water through because it keeps them from getting the guinea worm transmitted to them. So the reason that works is because if we look at step 1 at the top here, if a human being drinks unfiltered water, these copepods contain the larvae within them. But if you drink filtered water, the copepods are too big to get through the filter, so they get stuck outside. So if you drink unfiltered water, the larvae are released when the copepods die in your gastrointestinal tract, and then once they are released, they penetrate your stomach and intestinal wall where they mature and reproduce, and then fertilized female worms migrate to the surface of the skin. Remember, this takes about a year, and once she's ready to release her larva, the female worm moves toward the surface of the skin and causes a blister. This blister typically burns and makes the person want to put their foot in water, and as soon as the blister is put in water, the blister bursts and the larvae are released into the water from the worm, and the cycle continues because the larvae are consumed by the copepod, and then the copepod can be consumed by the person. So 2 avenues of stopping this infection. Number 1, drinking filtered water. So it doesn't even have to be chemically treated, it just needs to be filtered. Now, of course, that's not going to change a lot of other things that you might contract from contaminated water, but it will keep you from contracting the guinea worm. The other area of this eradication campaign is the release of the larvae. So they don't release haphazardly. You feel them getting ready to release. They're typically released all at once. So all you have to do is put some water in a bucket and put your foot in the bucket, and you will release the larvae into the bucket rather than releasing them into the water supply. So it's a dead end for that female worm because now she's released her larva into a bucket, that water can be sterilized, and you've stopped transmission of the guinea worm. So, gross disease, but good news, right? This infection is one that is moving toward eradication.